have the great privilege and honor of introducing Dr. Bruce Luxon, my boss, professor of medicine, chairman of the Department of Medicine. Just a little bit about his career. He has gone and followed the triple threat of academics, combining his clinical roles as a transplant hepatologist, and he's often traipsing back and forth to clinic. He's been an innovative educator and a nationally recognized administrator. He's been active in research. Dr. Luxon has a combined MD and PhD from the University of Iowa, where he completed his dual degree at the University of Missouri, Colombia, with his PhD in mathematics. He remained on the research faculty at the University of Missouri before undergoing his residency in internal medicine. He completed his training in gastroenterology and hepatology at the University of California, San Francisco. His research has been focused on chronic liver disease, intrahepatic transport of bile and fatty acids, hepatic fibrosis, and decision-making in the care of patients with hepatitis C and those in need of a liver transplant. He's had over 25 years of experience in clinical care, especially for those patients who've been suffering with hepatitis C. He has considerable experience in treating those with hepatitis C and HIV co-infected patients with the additional comorbidity of hemophilia or other bleeding disorders. He serves in many leadership roles, including being our departmental chair, also leadership roles nationally with the Association for the Study of Liver Diseases, American College of Gastroenterology, and the National Hemophilia Foundation. We are so lucky to have him. He's been so supportive of everything that we do in education, but also has been a great leader in all of our clinical and research missions. So with that, I will introduce the boss, Dr. Luxon.
Thanks, Sean. I'm not sure I can live up to that introduction, but I'll sure give it a good try. So often in November, I try to give an update on clinical things that I think are changing rapidly, and that's what today's talk is going to be. It is not about COVID, for those of you who are worried about that. Instead, it's going to be about eyes, muscles, lungs, and of course, livers. And I'm going to talk about gene therapy for diseases that are in each of these, and I'll show you why I'm interested and have been interested in these as I go along. Do have some disclosures for you. I do work for 2 pharmaceutical companies as a data safety manager for Hemophilia Gene. And as Sean said, I'm the sole hepatology member for MASAC, which is an unpaid but very gratifying position on the National Hemophilia Foundation. And you'll see why I'm interested in that in a little bit. So you might say, what's with this title? I was expecting something, that I would know about. Well, we're going to talk about eyes, muscles, and I'll come back to see how many of you recognize somebody with the muscles. Going to talk about lungs and finally livers. If you look carefully up in the top right corner there, the middle individual is Sean Connery, who was discovered at a muscle building contest. I don't think he's got that many great muscles, but there's Sean Connery many years ago. As you know, he recently passed away. So we're supposed to start with a case, and so this is one of my patients that I see occasionally or saw occasionally. He's a 48-year-old male. He has hemophilia B, and he came to my care because, as Sean mentioned, he had hepatitis C. He got it probably in the 1980s when he was using factor concentrates. He also got he also got HIV. I'll show you a picture of his knees. I was able to cure his hepatitis C, but I did not have a cure yet for his hemophilia or his HIV. And he switched to prophylactic infusions about a decade ago, which is now one of the standards of care for patients with hemophilia. And this individual really wanted to be stopped being referred to as a bleeder. In fact, he wrote a book, which I've shown you on the bottom left there, entitled The Memoir of a Bleeder. And you can see his knees on the bottom right there. when the bleeds occur into his knees, obviously they damage the joints, and you can see how terrible his knees were when I first met him. He entered a trial with gene therapy at a large university hospital, one of those so-called academic medical centers that is located in one of the Carolinas, and I'll let you figure out which one that might be. Occasionally, I try to make sure that at least somebody stays awake, so I will show some humor slides for you. Here is my first attempt at that. This is indeed a sign that says if you hit this bridge, if you hit this sign and then turn right and go over the bridge, you will take out the top of your semi. So there's a pretty large sign. You'll see those occasional humor as we go by. So here's what I'm going to talk about today. I'm going to give you a brief background of what gene therapy is, including which diseases might be treatable by gene therapy. I think you'll be impressed how many there are. I'm going to talk about how it works and again, emphasize why I got interested. I'm going to go over 4 examples of diseases that have been attacked. They happen to affect the eyes, muscles, lungs, and liver. Talk briefly about animal studies, but mostly about the human trials. I want to impress on you the successes that we've had, but you also see some failures, but we're really making progress. So if we were in person, I would call on some of you to tell me if you've ever heard of this particular disease. And when I started with gene therapy, I thought that they were all really rare diseases, that, you know, they were storage diseases. They were 1 out of 100 million diseases. But as I go through this list, and these are the ones we're going to talk about today, I hope you'll appreciate how devastating these diseases are and actually how common they are. Muscular dystrophy, I bet you most have heard of. I hope all of you have heard of cystic fibrosis. And as I mentioned, hemophilia is going to be my 4th disease that I talk about. So there are many rare diseases for which we know a genetic mutation that causes the disease, but we're not sure yet how to correct that. But I'm going to argue that there are also many common diseases that have genetic causes that are well understood, and maybe we will be able to cure these as time goes along. So what is gene therapy? It's a process in which a malfunctioning gene is just replaced or counteracted. What I'm going to show you today is when we replace it, but there are other examples where we try to counteract the malfunction gene's product. We want to target cells that are adversely affected by the malfunctioning gene. That's one of the harder things to do, and I'll show you why it's particularly difficult in cystic fibrosis. We use gene transfer to deliver a new gene, a new genetic information into the target cell. And then these gene-corrected cells, if you want to use that term, must be present in large enough numbers to reverse the condition. So if I affect 1 liver cell, for instance, that's not going to be good enough. I have to infect or transfect many, many ones. And these cells have to live long. They have to derive proficient expression of whatever therapeutic molecule we're trying to replace. And we can't mess up the rest of the regulatory mechanisms in the organ. So you might say, well, is this something that is widespread? So I guess a week ago, I looked on Clinical Trials Gov website, and I was stunned. There were 635 trials listed using the code words gene therapy. 214 had been completed. 182 were still recruiting or enrolling. And the topics were, as I mentioned, the storage diseases, hemophilia, some vision diseases, which I'll show you today, cancer, Alzheimer's, muscular dystrophy, and my current favorite topic, aging. So I am accepting donations to try to stop me from aging anymore. I've shown you at the bottom right a pamphlet that comes out. This is part of the humor section, folks. Curing aging, as you can see down at the bottom, if you pay $1 million, you can go to a clinical trial in Colombia, located in South America. and they will cure you from aging. So anybody who's interested in donating to my fund, let me know. Of course, a hepatologist would recognize that the first gene transfer had to do with the liver. And we discovered that many years ago that certain liver diseases would go away after we did a liver transplant. But we noted also that this is quite a wasteful form of a gene therapy, because tens of thousands of the normal genes were modified, including on a couple of cases, we caused a genetic disease. It does correct a single defective gene, but it involves a moderately invasive surgical procedure. So not quite the way to go. But we did recognize that hemophilia and the porphyrias, which are two diseases due to liver issues, were cured by a liver transplant, but not the way to go. So if you're looking out, what diseases should I focus on if I'm going to become a gene therapist? Well, first of all, you got to know the gene that causes the disease. And I think we've made huge improvement in that. We must know how to improve the gene. We must know which cells are important to target, and we got to have a way to get to those cells. And when I come to cystic fibrosis, you'll see that we failed in many ways on that criteria. Here's some recessive gene disorders that we're working on: cystic fibrosis, hemophilia, muscular dystrophy. I'll talk about all of those. And finally, fixing the gene must fix the disease as well. So many diseases are possible, but more challenging for some. So this is one of my first cartoons. So all of these studies that I'm going to show you today use a virus, a special virus that is made in the shop, so to speak, to create a good virus that transports the genetic material into the cells where we want to make the changes. So we start off with a HEK cell, which is a human embryonic kidney cell that's been around since the 1970s. And it's an easy thing to transfect with a virus. So we start there. We have a gene that we want to put in. We then make this cell, which can be grown in huge quantities. We make it wholly so that the material that's outside the cell can slip in. There's the holes. And then we let the virus that we've made get into this cell. And what it does, the cell very nicely turns out a recombinant that now has the new gene that we want, also makes a few proteins, but then you can separate those and you have a virus now that includes the genetic material that you want to transport. So knowing that this HEK cell is a way to go has made this very easy in many ways to do. And I'll show you some examples of how this has been very effective. So as you're making decisions about that, you need to know, how much stuff do I need to move? Am I going to be able to get by with a pickup load? Or is the gene product that I need to move going to require this big machine on the right? And again, when I come to cystic fibrosis and muscular dystrophy, I'll show you how we've gotten much smarter about that. Obviously, you need to know a truck versus a moving van. And then you have to get the package, the virus in this case, to the right cells. Here's a map I pulled off the internet for a Tokyo subway map. I would hate to negotiate that. This one you probably recognize is a little more simpler. But again, the key that I want to emphasize is you have to know how to get this virus into the right cells. You also need to promote the product. And by that, I just have some cartoons that I'll go through. But if you get the virus in the cell, you also need to make sure that the cell is turned on to make the protein or the enzyme. And so promoting the product is really good molecular virology. And I'll show you some examples of that. That's way above my head, but promoting the product is very important as well. So what do we package in this vector? Well, we often just go to a process that's already been used before. What's up at the top of the slide is a copy of how we make one of the recombinant factor 8 in a CHO cell line. But I just copied that out. So we know how to do that. What we do then is package this gene shown at the bottom in the green into this virus. And so we already know how to make the gene and transfect it, if you will, into a hamster ovary cell. Now we just put it into the virus. And making that is really an important concept. But if we've done that to make just the protein, all we're doing now is taking it from a reactor in a biology lab into the human body and getting us, the human, to make it. So I've used the term AAV a couple of times already. It stands for adeno-associated virus. Why do we use that? Well, it's naturally occurring. It's easy to work with. It's pretty small. There are different serotypes out there, and I'll show you some data on that as we go along, because that makes a difference where the virus tends to go, and we think it's non-pathogenic. It's also easy to package stuff into. It doesn't replicate, it doesn't integrate, and as I mentioned, it has a good safety profile, both in non-clinical as well as clinical studies. So it's an improvement. It's an easy vector to use. Now, I'm not sure where this sign was found. It says, beyond this point, you may encounter nude sunbathers eating waffles. I'm not sure where that is, but if anyone has driven by that, please let me know. And I think I'd fit right in. I bet you very few of you know this answer, but I'll give you some hints. The first FDA-approved disease treated with gene therapy had to do with the eye. Now, this is an age issue. I'm hoping that some of you know that this is Marty Feldman, who was a British actor that appeared in comedies. And of course, as you look at those pictures, you can tell he was best known for his exophthalmos. So the answer to the question was a retinal gene therapy. So we're going to switch now from kind of the basics about what gene therapy is. I'm going to give you 4 examples of diseases that we've attacked using gene therapy. So we know that there are over 250 genes that have been identified in these inherited retinal diseases. The genetic mutation causes a protein or an enzyme that's absolutely essential to sight to be misproduced. And so blindness develops, often in pretty early childhood to late adolescence. It's a slow and progressive loss of sight. And one of the diseases that I'm going to talk about is a form of retinitis pigmentosa. It affects 1.5 million people around the world. It's not that common, but in it, we've found that there's 90 to 200 different genes, and we're going to talk about one of them today. The gene we're going to talk about is the RPE65 gene. It makes a retinol ester isomerase, and that's an enzyme that's crucial to retinoid recycling within the eye. If you don't have this enzyme, the rods get damaged, the retina gets damaged, and blindness appears. We've done animal models using dog and mouse, showing that replacing the gene with a functioning one corrects this biochemical blockade. And so phase 1 trials in humans came along. They worked pretty well, and I'll show you some of those. And then larger trials ensued. So why would they use the eye? I was stunned until I thought about it a little bit. But the retina, where we're going to inject this material, is pretty isolated. It's immune privileged. It has a blood-body immune system isolation, so that's good. You don't have to have a lot of vector because you're putting it in a pretty small space. It's relatively easy to access the eye. I'll show you some pictures, not ones that I would recommend during the lunch hour, so they're cartoons rather than the actual pictures. And the cells that we're going to try to transfect are stable, meaning they're already differentiated, they're not dividing, so if we're able to fix them, the fix would be permanent. Again, a very important feature. So what was the first approved product? It was one of these adenoviruses The subtype was 2, and it contained this enzyme's DNA. We ended up using a promotion. As I mentioned, it was important to make sure that the cell that we infected, transfected, turned it out. We used a chicken beta-actin promoter with a CMV enhancer, whatever that all means. It means that when we get it in the cell recognizes the virus, the vector, uses that DNA, and really turns out a lot of the protein. So I have a couple of pictures to show you where this protein is. As you can see on the slide, when light comes into the visual cycle, it starts, we end up at the bottom of the slide forming a retinal ester. This protein that we need converts that to a cis retinol, and that's very important. If you don't do that, you can't respond to light, you can't regenerate your retinol, and so eventually you go blind. And then this just again shows you on the right-hand bottom slide, in comes our vector at the top of the cycle. It gets into the cell. It's then taken apart. The genetic components gets put into the cell. The cell starts to make the protein, and everything else then goes forward. How do you get it into the eye? Well, there's two ways to try. This is my cartoon for you. One is to put it right into the bottom of the retina. That's shown at the left. Requires a little more fancy surgery to do, but ophthalmologists are good at doing this. The other is to just put it into the vitreous humor, but the studies I'm going to show you relied on putting it at the back of the retina. Again, why? That's where the cells are that we want to transfect, and you don't have to have very much product to put it there. It's not wasted. So here's what the ophthalmologist does. They put a couple of probes into the eye, and they take out the fluid. They then insert, as shown in step 1 there, and make a little bleb underneath the retina. They then inject into the bleb the vector that we want to put in, and then they check it to make sure everything's fine. So it's pretty simple. I'm going to skip this one. The first injection was done right here. I found a picture of this, and I'll tell you why in a second. It was done at the University of Iowa. We had a big retinal ophthalmology program. So many of the studies I'm going to show you were done at my old alma mater. Take about 10 minutes for this to occur. So we started off with animal models, as I mentioned, and at the end, the animal was sacrificed, and we'd measure the enzyme, but that's not really quite applicable to humans. So the Iowans, rather than using a cornfield maze, developed a different kind. It's a multi-luminescence mobility testing, fancy word for a maze. It's just a 5-foot by 10-foot obstacle course. And in it, there were steps and doors and some obstacles. And we'd have the patient, before and after the therapy, walk through this maze at different levels of light. And if a normal sighted person could get through the place easily, not make any errors, and I'll show you a diagram of one, at a very low light level. Affected persons really had to have more light because their vision was affected. So here's the project that happened. The worst eye that was most affected was injected first. If there were no side effects, we waited 6 to 18 days. Then we injected the second eye. We did safety checks at 1, 3 days on the first eye, and then 1, 3, and 2 weeks later. And then we had the patients go through the maze after the therapy at, look, 30, 90, and a whole year later. And here's kind of what the maze looks like at the bottom. You start at the very bottom right, and there was a hole to the right, so you had to go forward, miss the form, not fall into the next hole, and follow those areas, going up some steps, going around some more holes, avoiding some grass, and then finally getting out the door. And what we measured was, did you run into those obstacles? So here are the results that I wanted to emphasize for this. Let's start with the line in blue. First of all, this is their how well they did on their walk through the maze. And at the bottom of the graph is, again, 30, 90, 100, and a whole year later. You can see that the control group that did not get the therapy really didn't get much improvement. But look at the green bars. There's the control group, no improvement. Look in the green, they got by at nearly maximum improvement. So they could walk through there at the lowest light level that the light had. So they're back to normal sight, and it stayed. Notice that it wasn't a transient issue, but over a whole year, these children were able to see again. Pretty impressive, I thought. So here's the first drug. As you might guess, it's a little expensive. It was $850,000 to cure a rare blindness disease. The product's name is Luxturna. If you're at a trivia contest, a Zoom trivia contest, that's what it's called. And this was done in 2018. So you think, my goodness, that's a lot of money. But think about saving people who are in their grade school, who are going to turn blind. They get one shot and their blindness goes away for life. That's a pretty good bargain. So did anybody feel like this when they got their COVID testing? Alicia found this on the website, and certainly my experience when I had my first COVID test. All right, I'm done with eyes. I'm going to move to muscles. Again, I hope many of you would recognize this individual. It's Jerry Lewis, who for many years as I was growing up did MDA telethon. And so the next disease we're going to talk about is muscular dystrophy. It's a disease that I think most of us have heard about, partly because of the telethon. It's an X-linked disease that affects boys, therefore, but it's fatal, and I'll show you why in a little bit. This gene that's defective codes for a protein called dystrophin. And dystrophin, I'll show you a bunch of diagrams about that. But what I want you to remember is that it maintains muscle stiffness, and it really protects the muscle against the stress of movement, and it anchors the cytoskeleton. If you don't have this protein and your muscle moved, then every time you move it, there's a contraction injury. And this is a problem in skeletal muscles, it's in the diaphragm, it's in the heart. So you can imagine that this is an overwhelming disease to have. So here's some milestones for those of you like me who had kind of forgotten some of these milestones. So it starts off, you have motor delay, and people notice that in children 5 to 7 years. Pretty soon by their early teens, it's loss of ambulation, they're in a wheelchair. By their teens, the diaphragm's affected, sometimes their heart's affected. And then look at the right. In the teens to 20s, people die. So again, a very disturbing progress in a disease that hopefully we can cure. I'm going to start off again with some mouse experiments. These were using one of the vectors, an adeno-associated vector, and it's a serotype 2. Start up at the top, it's a wild type, and this is what the muscle looks like when you stain a normal mouse for the protein, plenty of it in the green. The second box is pretty dark, is a knockout mouse before treatment, no green. And then here's at the bottom are 3 pictures of a heart, a quadriceps, and the diaphragm after we transfected the mice 6 months later. And look, you can see plenty of the protein. So it's like, wow, this is a huge success. There was very little T-cell immune response. That's great in the mice. So it's like, yes, let's move it on to humans. So excuse me, we started human studies. These were reported now a decade ago. We had 6 patients that got injected with a very similar vector that we had used for the mice. But the problem was nobody expressed any of the dystrophin. The protein that we wanted didn't show up in any of the muscles. And we had to figure out why. It turned out that half, in contrast to mice, had immune response against the vector that we were using. All of the patients developed neutralizing antibodies. So as soon as we put the vector in, the virus in, the antibodies took it out. So we quit. There are no further clinical trials with this product. Again, an example that, wow, mice, it works really well. Humans, disaster. 2010. So I put up the next couple of slides has some cartoons that just show you the brilliant science that I think has gone on about muscular dystrophy. So this is a schematic of what we think the full length, the normal protein does. Let me just focus you first over here. And so part of the protein hooks up and provides a connection, if you will, to the sarcolemma. You can see some hinges there, and the other binds the F-actin. So that part looks pretty important. Now look in the middle as you go from left to right. Just doesn't do much, does it? And then finally you get over to the right side, and there's a bunch of more things that seem to be more important. So the protein structure was very helpful as we went through this. Now here comes the lucky part. So it turns out that there were patients who were much older than the teenagers that I told you about. And so here's a single patient who was 61 years old. He had really mild muscular dystrophy. He was still walking at age 61. And when they looked at his protein, they found out that it was different. Look how short it is. And yet, here's the part that interacts with the cytoplasm and the F-actin. Over here's the part that interacts with the microtubules and stuff. And then underneath, we, as you can see, was, oh, here's the parts that we need. That's the active part. We don't need all that middle crap. All we need is the active part. So some very smart people did some science. This was from the Nationwide Children's Hospital. They made their own microdystrophin. They made their own protein, knowing that only parts of it were really needed. And so it looks very similar to what this 61-year-old had. They maybe made a few small changes, but they decided, you know, maybe this is what we need. This smaller protein will do what we need. So there was an intensive effort to, again, design a smaller but a functional cDNA. As I mentioned, the full length was too big to be transported and had all sorts of trouble. The missing middle really wasn't needed. And so the scientists made the smaller thing, the micro dystrophin that you see at the bottom. So then it was time to develop this. And so went back into the humans. 4 children, again, these are pretty young. The average age of these 4 kids were 4.8 years. They got a single dose. It was an IV infusion. And the vector that they used was really unique. Again, these are very, very smart guys. It was not the typical adenovirus. This was one that they found in a rhesus monkey. So I'm not sure how they found it, but it was a new version. It's a virus that likes to go to skeletal muscles. Great. It has a minimal human immune response, much different than the first studies I showed you. They were able to put into this virus a very special promoter that's found in skeletal muscles, including the diaphragm. So again, very hot science. And then they even further modified the virus with what they called an enhancer that drives expression in the heart muscle. So those things that I've marked with asterisks are just fantastic basic science. So just hot off the press 2 months ago were the first trials of these reported in the JAMA version of Neurology. And the first things that they wanted to do was make sure it was safe. So these are some muscle biopsies. The top 4 of the 4 patients show just pre-treatment, and the bottom 4 show post-treatment, and the muscle biopsies look normal. Good, so it looks safe. Then they did some more staining, looking at, again, before and after muscle tissue. Looks very safe. So safety issues, not a problem. Good. And now did it work? So let me walk you through this one. At the very far left on the top is a normal level, and you can see the red staining of the protein, dystrophin staining. Look at go to the right now on the top, all black. Those are the 4 patients' gastrocnemius's muscles before treatment, and now go down, and you can see there's the protein. So it's very effective. I think these were 6 months after the single IV injection. So dystrophin is now produced by the muscles. Oh yeah, so normal pre-treatment and then post-treatment. Now, besides the staining, they also wanted to make sure that it was the protein that they had put in. And so these are Western blots. That's how you measure protein. And I just highlighted with the red arrows that this is really this modified microdystrophin protein that's expressed in the patients. So there it is. And did it work? Did it make any difference to the kids? Well, the CPK levels, which are normally very high, again, because the muscles are being damaged, dropped very quickly from 27,000 on an average to only 8,000. Some functional things also improved. There's a time to rise test, whether they can climb 4 stairs, how far they can walk, or how long it takes, I should say, to walk 100 meters. And all of the kids improved, but how much they improved depended on where they started off. So it depended on the severity of the illness, but all 4 kids were much better and no safety issues. So huge, huge step forward. So I think it's ready for prime time soon. Again, this just came out in 2020, 2 months ago. You watch, we'll be having more studies for muscular dystrophy. Switching gears, this is before COVID. This is one of the signs on my way home that you have to know exactly what time it is, what day it is, and you have to read really quickly when you have to go only 25 miles an hour. My wife, who's watching, knows that I failed this one time, and one of the electronic speedometers caught me going home at 30 miles an hour, and was nice enough I got to contribute to the DC Traffic Violation Fund. Very nice. So we're going to move on to cystic fibrosis. So we've known about cystic fibrosis for a long time. I just put this up. Again, it's a defect in one of the channels, and that we've known about since, I believe, 1989. They're different channels, but it's one of those diseases that affects both the lungs, but also the liver, and so we've known what the defect is, so it'd be a logical thing for us to try. The next 3 slides just show you kind of a time course of what you're going to see is not so successful. So as I mentioned, we found the gene, 1989. We thought, oh, good. We can put in some new genes, and so we started that in the mid-90s. 93 was the first adenovirus clinical trial. They all failed, and so by 1999, a mere decade after we found the gene, we quit. Wasn't working. So we had to try something different. Then we switched viruses. We went to the adeno-associated viruses that I've been talking about in 1999, went through some more work. but they didn't work either. So by 2007, we'd quit. So the therapy that I just shown you that was so successful for retinal disease and will be so successful, I think, for muscular dystrophy, failed miserably in cystic fibrosis. And so we started looking for a different virus to try to deliver the product to the airway, which is where we thought it needed to be. And through a series of, I think, again, really brilliant work, we switched to something called a lentivirus. I took this slide from a paper called Preparation for the First Lentivirus Clinical Trial. Came out in 2018. So they've done the animal studies and they're close to getting ready, I think, to trying it in humans, but I did not find any evidence of that yet. So I did want to show you some mouse data. These are not the change. This is just trying to get the vector into the right cells. So remember, cystic fibrosis can affect multiple different organs. We're trying to make sure that we get it into the correct lung airways. So this is looking at a simian virus. On the left, we've tacked the simian virus onto a protein that glows called GFP. You can see the green glow. You can see on the right, no green glow. And then if you look at the bottom, you can see in these mice models that we do get the gene product, in this case, not the one we want, but a fluorescent protein into the mouse lungs. Good start. Then we did the same. We put in the real gene and then gave it again to the mice. And you can see on the right-hand side that we got by day 7, we got a lot of the protein into the airways. Notice that the scale is logarithmic. So it goes from essentially none up to thousands of measures of the protein, and it stays there by day 28. Making progress in mice. So what does the future hold for these? We're going to switch viruses. We're going to use a lentivirus, and it looks like it's the best one because it gets into lung tissue. It doesn't have the immune response that our previous trials had. And at the moment, if you read the literature, you're seeing that they're trying to get better promoters and get better gene products delivery. But as the data I just showed you in mice, I think we're there. So pretty soon, I think we'll be coming along to actually start the new next series of experiments for cystic fibrosis using the lentivirus as our carrier. So I'm going to finish up back where I started with the case. Again, I wanted to show you some knees from patients that have hemophilia. Here's Shelby's book again. So he had hemophilia B. It's characterized by a deficiency in factor 9. It also is named as Christmas disease, not related to the holiday, but to Stephen Christmas, who was a patient who had it. It's also known as the royal disease. You probably have heard of Ryan White. He also had it. And we now know that there's more than 100 mutations that we've identified in the gene that causes hemophilia B. So as I mentioned, patients end up taking factor. We used to use factor obtained from humans. Unfortunately, we gave hepatitis C and HIV to patients, and I mentioned Ryan White. We now have recombinant factor, but you have to give it fairly often to keep from having bleeding, and so it's a pain. It's also very expensive. So why am I interested in it? I got involved to start with because of the hepatitis C, but also now gene therapy we know can cause an immune reaction, and I'll show you some slides about that. We know a lot about how the vectors get into the liver. I just put this in as you must have a colorful cartoon, but I made my own. So here's the steps of gene therapy just from the liver. So there's a very nice liver cell that you can see in the middle. There's our vector that we're going to use, with the red material being the product we wish to deliver. It gets into the liver cell, and on the next slide, I'll show you how we do that. After that, it dumps off its vector DNA into the nucleus. The capsid's still around. The capsid's torn apart by the liver cells, making a bunch of peptides. And then the liver cell doesn't like those peptides because they're from a virus. So they stick it onto an MHC complex, stick that on the liver cell and try to get rid of it. Unfortunately, there's some T cells that come along and they see this complex, they hook up, and now they go, this is a liver cell that's infected with a bad virus. I should kill the liver cell. Uh-oh, that's the wrong answer, but that's what happens. And so that cell that we've now successfully transfected and will be making the product that we want is killed. So it doesn't work out so well. There are multiple different ways that these AAVs can get in, and I've shown that on the left-hand side of the slide. The different serotypes all have different ways that they can get in. So we're not exactly sure which one is best for the liver, and we've tried multiple ones, mostly AAV2 and 5. They all get into the liver. Why would it be good to get the liver done or transfected? It's because the liver's, like the chairman, often resting. And so they're quiescent. So if you can transfect a liver cell, the hepatocytes will exist for many years, and you should have a long-standing production of the new gene product. So our goal, of course, is to increase the factor, minimize the need that the patients have to take factor. And we have multiple companies, 2 of which I mentioned in my disclosures, that are working on these products. I'm just going to give you a couple examples as I close up. We again started off with mice. We looked at the different serotypes, and it's really easy to determine expression in animal models. And the different serotypes tend to go down the sinusoid at different rates. And so I just put another colorful picture in for you. On the left, the serotypes 2 and 7 tend to go around a central vein, which are shown by the red arrow. A rhesus version and serotype 2 is somewhere else. It's more uniformly distributed. I'm going to skip this one. Let me just go through it real quickly. So the work in mice really worked well. And so again, it's time to try in humans. Let me show you that. So here's 2 or 3 examples of a treatment with humans. And what I show you on the vertical scale is the factor activity. And on the horizontal scale is the time after a single injection. And you can see there's 5 participants there. So higher levels are good. Let me just highlight that these patients all started off with bad disease, severe disease, which means that their FIC level, factor 9 level, was less than 2. And all but one of the patients got a great response. This is cohort 1. That was a low dose. Then we increased the dose, and you can see everybody got above the kind of critical level of 2. Some had quite a bit. So it looks, wow, this is great. So the protein's there. What happened to the patients? You can see on this slide that they were using a lot of factor replacements. The 2 cohorts are shown there, the blue, the low dose, green, the higher dose, and then in orange, we added those up. But look what happened after we gave them the treatment. Their reduction of factor their use of factor was greatly reduced, almost non-existent compared to before. And did they bleed? And the answer was no. Even with reduced use of factor, in some cases 0, they still didn't have any bleeding. So wow, 1 single shot, don't have to infuse factor, and you don't bleed. I'll let you read that on your own. Animal prevention, cruelty prevention. Let me skip that one. Hold on. So one of the trials, and this is my last trial I'm going to talk about, we wanted to make sure that it lasted. So this paper just came out again this year in the New England Journal, and it followed patients for 3 years. This is not factor 9, this is factor 8, but this was very important, I thought, because I thought this was what we needed. There were 2 doses of vector that were used initially, a low and a high. No safety events. There was a mild irritation of the liver naturally, but that went away. And this just shows you the factor 8 levels. And notice that this goes out to 2 years. The different colors are just different assays. So just notice that all the bars are way above 2. So there's plenty of activity, and it persists for years. 2-year follow-up. Here again is, did they need to use factors? And you can see before the infusion at the far left, they were using quite a bit. 3 years afterward, almost nobody's using it. I'm going to skip that. This is my last attempt at humor. I, again, attribute this to my wife. If attacked by a mob of clowns, go for the juggler. Chuckle. So, my 2 summary slides. I hope I convinced you that gene therapy for many diseases is now possible. I've tried to give you 4 examples where we've made extreme progress, 2 of which I think are prime time. In fact, one's FDA approved. I've talked to you a little bit about how to choose the virus vectors. And I've shown you that gene therapy can be used for many diseases and given you some examples. For all of these, we use animal models to start with to try to design the vector and look for toxicity. And I've been stunned how many human trials are different from our animal models. And even the non-human primate models don't always predict human results. But nonetheless, we've made considerable progress. We've used different AAV vectors. We've made some custom ones. We've made custom lentiviruses. And long-term results for some disease are disappointing, but I'm really optimistic that progress is being made. And for many diseases that I think we can attack, the long-term results are very promising. And the FDA has approved several gene therapies already, and I've shown you one of them. And with that, oh, my crystal ball. So my prediction of what's going to happen is that many of these trials that I've shown you will be out there for more common diseases. Cystic fibrosis, muscular dystrophy being the most promising. I think you're going to see that they're going to cost a lot of money. But on the other hand, if you can prevent the morbidity and mortality by a single infusion that works, that has no side effects, it's a huge improvement. And with that, I thank you, and I'll be glad to answer questions if I can figure out how to read them. Thank you very much.